Another area of nanotechnology that's really intriguing is DNA computing. And what's going on here is that researchers are building computers, because again, DNA harbors digital information, using DNA and the components of the cell's machinery that manipulates DNA. And the reason why they're able to do this is because when you look at what's called DNA metabolism, processes like transcription, where the cell's machinery copies the information in DNA and then locates it to another portion in the cell for use, or when DNA is replicated, or if there's damage to DNA and it is repaired, these operations are fundamentally the same operations that essentially are at work in a computer system. Because these machineries function like a biochemical Turing machine. So I'm not going to get into the details of that. But a few years ago, when Leonard Adelman, who is a computer scientist at the University of Southern California, realized that relationship between computer operations and the metabolism of DNA, he had the brainstorm of actually building computers using DNA. And these computers are held in little tiny Eppendorf test tubes that are about that big, but they're more powerful than the most powerful silicon-based supercomputer system. And part of that power is the possibility of doing massive parallel operations. And again, it also has to do with the massive amount of data that DNA can store, and these operations are operating at near maximal energy efficiency. And in fact, there are problems that you can solve with DNA computers that you cannot solve with traditional silicon-based computers. So, again, DNA's optimal nature is inspiring technology. Another area where DNA is inspiring technology is in nanoelectronics, where people are trying to build electronic systems at a molecular level. And one of the properties of DNA that's really, really unusual is that DNA can serve as a wire. The DNA double helix can function like a wire. And this was discovered in the 1990s by a scientist by the name of Jacqueline Barton, who was at Caltech. And what she did in this experiment is she had a DNA double helix, and then on the ends of the double helix, she attached two chemical groups. And these groups could give up and receive electrons. And so what she did is she hit one of those chemical groups with light that caused it to eject its electron. The electron flowed through the interior of the DNA double helix like a wire, and then it reacted with the other compound at the other end, and that compound picked up an electron. And the conduction through the DNA double helix was so fast that when she originally reported the data, people didn't believe what she was describing. And it turns out that actually the electrons move through the DNA wire employing a phenomena called quantum tunneling. It's really, really interesting. Well, as soon as Jacqueline Barton made this discovery, people thought, well, DNA could be used to build nanoelectronics. And a few years later, Jacqueline Barton's group discovered that the cell actually uses this mechanism to detect damage in the DNA double helix. Because when the DNA double helix is damaged, the helix is distorted. It's like breaking the wire, and the electrical current doesn't flow. And so what the cell does is it surveys the entire genome with these proteins that bind to the DNA. When they bind, they give up an electron, they send it through the interior of the double helix. Another protein that's bound to the DNA will pick up that electron, it'll hop off, move down the DNA double helix, bind again, and send its electron. And so you have these proteins just sending electrons back and forth through the interior of the DNA double helix. And if there's damage, it's going to cause the double helix to distort. And when that happens, the electrical current flow stops, and these surveillance proteins begin to stack up in that particular region of the double helix, which triggers a DNA repair process. And again, this idea is inspiring work in nanoelectronics, where people are looking at the DNA double helix as a wire molecule. So the point here is this, that DNA has such an optimal design in terms of its role of data storage that it's literally being used, again, to inspire new technology. And this is really provocative, but what's even more provocative is I believe that the area of biomimetics and bioinspiration actually can lead us to a new set of arguments for a creator's existence. The fact that DNA inside the cell is manipulated by the cell's machinery in such a way that it resembles a computer system, so much so that people can take advantage of that and build DNA computers, suggests that these systems inside the cell must be the work of a mind. This is what Leonard Adleman says, the founder of DNA computing. The most important thing about DNA computing is that it shows that DNA molecules can do what we normally think only computers can do. This implies that computer science and biology are closely related, that every living thing can be thought to be computing something, and that sometimes we can understand living things better by looking at them as computers. It's practically stating the same thing as William Paley. Now, when I wrote the book, The Cell's Design, where I argued that there is this eerie similarity between biochemical systems and man-made designs, making a modern-day version of the watchmaker argument, one of the critiques that came from skeptics is that, well, this isn't really science because it's all well and good to make this argument, but it really isn't science because you can't really make any predictions using the watchmaker argument. And so to counter that criticism, I actually presented what I call the watchmaker prediction in the cell's design. And the idea behind the watchmaker prediction is this, that if indeed the cell is the product of a creator's handiwork, that there's very well technology inside the cell that is anticipating our technology advance. In other words, we have to wait till we develop new technologies before we realize that those very technologies already exist inside the cell. We just can't recognize it as such because we haven't invented the technology yet. And it turns out that DNA wires are a beautiful fulfillment of the watchmaker prediction, because the book, The Cell's Design, was published in 2008, and Jacqueline Barton and her group didn't discover the fact that DNA wires are used to survey for DNA damage until about 2012. So what Jacqueline Barton and her group did is discovered this property of DNA that allows it to conduct electrical current, and then it suggested that this could be used in nanoelectronics as a wire. But it was only later that they discovered that this is actually how the cell detects DNA damage. And so this is a fulfillment of the watchmaker prediction. We weren't able to even recognize this biochemical technology until Jacqueline Barton suggested the use of DNA for wires. Now there's another way in which we can argue for design that's a variation of the watchmaker argument, and this is what I call the converse watchmaker argument. That is, if biological designs are the work of a creator, then these systems should be so well designed that they can serve as engineering models and otherwise inspire the development of new technologies. And so the fact that we see, again, biomimetics and bioinspiration at play, again, highlights the elegant design of these systems. And we can, again, formalize that intuition of design in the form of an argument that I've dubbed the converse watchmaker argument. So these advances in nanotechnology inspired by DNA allow us to make three variations of the watchmaker argument. They also allow us to respond to the claim on the part of evolutionary biologists that the watchmaker is blind. This is an idea that was articulated by Richard Dawkins in his book, The Blind Watchmaker, where Dawkins is arguing that the designs that we see in biology are not the product of a mind, but are the product of natural selection, which if it's a watchmaker, it's a blind watchmaker. This is what Dawkins said. Paley had the proper reverence for the complexity of the living world, and he saw that it demands a very special kind of explanation. The only thing he got wrong was the explanation itself. The true explanation had to wait for Charles Darwin. Natural selection, the blind, unconscious, automatic process, which we now know is the explanation for the existence and apparently purposeful form of all life, has no purpose in mind. It has no mind. It has no mind's eye. It does not plan for the future. It has no vision, no foresight at all. If it can be said to play the role of a watchmaker in nature, it is the blind watchmaker. But I would actually argue that the discipline of biomimetics and bioinspiration represents a direct response in a challenge to the blind watchmaker claim. Why is that? Because the nature of the evolutionary process is such that it produces flawed or imperfect systems. Evolution does not produce optimal designs and elegant designs. It produces inherently flawed designs. Why? Because it's an unguided process, it's undirected, and it co-opts existing designs and has to cobble them together to create new designs. Ken Miller, a famous biologist who's involved in the creation-evolution debate, wrote this: "Evolution does not produce perfection. The fact that every intermediate stage in the development of an organ must confer a selective advantage means that the simplest, most elegant design for an organ cannot always be produced by evolution. In fact, the hallmark of evolution is the modification of pre-existing structures. An evolved organism, in short, should show the telltale signs of this modification." In other words, we don't expect evolution to produce optimal designs, but fundamentally flawed designs. That is, evolution produces kludged systems. Yet if this is the case, then why would anybody in their right mind as engineers turn to designs in biology or biochemistry to inspire technology? Why would you turn to a fundamentally flawed, imperfect design to inspire technology? That doesn't make any sense. In fact, it makes more sense to do biomimetics and bioinspiration in the context of a creation model or an intelligent design framework. Is it reasonable to think that biochemical systems represent the outworkings of an unguided, historically contingent process that co-opt pre-existing designs to kludge together new systems and then in turn use them to inspire new technology? And so I think what we see here with this whole idea of bioinspiration and biomimetics is a powerful response to the blind watchmaker challenge.